Welcome to tonight's webinar. Please welcome our moderator for this evening, Dr. Jackie Meinhardt.
Welcome, everyone. My name is Jackie Meinhardt, and I will be your moderator this evening. Our speakers tonight are Bonnie Crater and Nicole Bell. Tonight's webinar will consist of a 25-minute presentation from Nicole on the Center for Lyme Action's recent released report, The State of Lyme Disease Research in the United States. Following that, Bonnie Crater, our co-founder and board of director of the Center for Lyme Action, will present an update on the CLA's recent legislative activity. We will end with a 15-minute Q&A period. Please use the Q&A function located in your meeting control menu for any questions. You may submit questions at any time throughout this presentation. Our first speaker tonight is Nicole Bell. Nicole is an author, entrepreneur, and advocate for tick-borne and neurodegenerative diseases. She advanced her professional career as an engineer and program manager and spent the last 15 years in medical device and medical technology. Originally from Boston, Massachusetts, Nicole earned her master's degree in materials science and engineering from MIT and a master's of business in biomedical engineering from Duke University. She recently joined Galaxy Diagnostics as the chief business officer to commercialize advanced diagnostic materials or methods for tick-borne diseases. Let us please welcome Nicole Bell.
Thank you, Jackie. I'm going to share my screen. So it's a pleasure to be here, and I thank each of you for your passion and interest in the growing epidemic of Lyme disease. So a little over a year ago, I connected with the Center for Lyme Action, and Bonnie Crater, who you'll hear from next, asked me to take on a somewhat daunting project. As most of you know, Lyme has been a named disease for about 50 years. And in that history, there's been a lot of debate and confusion over some pretty fundamental issues that are affecting patients, millions of patients. And despite a lack of federal funding on the topic, there's been great research that's made key advances in our knowledge and our understanding of the disease. So CLA wanted a review of this research and to highlight key findings and critical questions needing further study. They also wanted this summary to be accessible to the general public, particularly members of Congress, so that CLA could use it as a vehicle to request more funding. So, many people I'm sure on this call know the space well, and they know that it's a very complicated space to navigate. And so that's why I described the project as daunting. But we leveraged a strong foundation and some fantastic experts in the space, and we launched the paper in July of this year. So I want to review some of the highlights of that paper today. But before I dive into the details of the paper, I find it's always helpful to start with the why. And the why here is that misdiagnoses lead to tragic outcomes. Like many people, I was blissfully ignorant about tick-borne illness for the majority of my life. Then all of a sudden it hit my family. My husband, Russ, changed right in front of me. He became moody and irritable, depressed. Eventually, I also realized that his memory was failing. And this, the man that I married was a brilliant electrical engineer and computer scientist. And all of a sudden, he couldn't remember the 4-digit code to our alarm system. We tested him for Lyme, but he tested negative. Only one of the 5 bands was positive on his Western blot. And after bouncing from doctor to doctor, he was diagnosed with early onset Alzheimer's disease, something that didn't make any sense because he had no risk factors or no genetic predisposition. 15 months later, we retested him and found that Lyme and other co-infections were in fact the problem. But at that point, he was in late stage disease and the treatments couldn't pull him out of his decline. And he passed away in January of 2022. Some of you also may have heard of Dr. Neil Spector. He was also a very healthy individual in the prime of his career, an avid trail runner, and then presented with third-degree heart block to the point where he needed a pacemaker installed to keep him alive. And even though he had one of the same infections as Russ, he had a completely different phenotype. And it just shows how varied the symptomology can be with some of these infections. Like Russ, he was tested for Borrelia and tested negative multiple times. Eventually, he did trigger the antibody test and got his Lyme diagnosis, but he needed a heart transplant because his heart had been so damaged by the bacteria. He lived about another 10 years, but passed away at the age of 63 due to the toll that the disease took on his body. So I'd like to say that these are isolated cases, but we know that they're not. Conventional knowledge says that about 500,000 people are diagnosed and treated annually. But these are the people that are diagnosed and treated as reported through insurance claims. That's how they came up with these numbers. But countless others work outside the system to get well. And there's the potential for millions more who are misdiagnosed. So it's a massive burden on the public and our healthcare system, and it needs to be solved. So the paper touches on a host of topics that I won't be able to dive into on this webinar, but I wanted to focus on the top 3 areas that I thought would be of interest to the ILAD audience. First is diagnosis. This is a big challenge, and I'm sure a lot of people on this call are well aware. Second, I want to highlight some of the research linking Lyme to chronic conditions, because that's a key area where we need more research. And then finally, there's treatment. I'll provide a brief overview of the challenges there and highlight some of the new approaches that are being advanced in the research lab. So one thing that's critical to understand about Borrelia is that the bacteria causing Lyme disease is that it's an elusive pathogen. It actively suppresses and evades the immune system so that it can survive within the host. So on the immune suppression side, it inhibits the complement system, the frontline immune defense of our immune system. It induces immune cells to release anti-inflammatory cytokines, down-regulating our immune response. It releases antigens to bind and neutralize antibodies to prevent the bacteria from being tagged for destruction. On the evasion side, it changes the expression of its surface proteins to confuse immune cells and prevent them from being identified. It swims orders of magnitude faster than immune cells, literally evading by outrunning the immune cells so that they evade detection and capture. And then it hides in immune privileged sites like joints, eyes, and the brain so that it can't be cleared. So the bacteria is highly adaptable and sophisticated, and over millions of years, it's figured out how to survive in a vast array of hosts, everything from cold-blooded insects and reptiles to warm-blooded birds and mammals. So why do I start with that as a discussion on diagnostics? Well, research shows that Borrelia has many, many ways to evade the immune system. Yet currently the standard of care of diagnostics is based upon what? Well, it's based upon antibodies. And this is our immune system's response to the bacteria. So not only do we have an evasive pathogen with a lot of variability, but we also have a massive amount of variability from person to person and how their immune system responds. So the result is massive misdiagnosis rates. And studies have shown that the standard 2-tier test and the modified 2-tier test that have been recommended by the CDC are only 40 to 60% accurate. So a compilation of studies is shown here, and that means about half of people will get misdiagnosed, which really isn't acceptable in any other disease state. And with in vitro diagnostics not meeting the need, doctors are left with rash presentation as a primary means of diagnosis for acute disease. But the problem there is that rash presentation also has a ton of variability. Study shows that rashes can vary drastically based upon sex, body location, age, ethnicity, and also some patients don't present with a rash at all. In the paper, we compiled rash presentation rates with the likelihood of a proper diagnosis of those rash types. And the analysis was based upon a series of studies that we reference here on the slide in showing what those prevalent rates and diagnosis rates are. And when you walk through the flow chart that you see here, it indicates that 52% of patients are likely to be misdiagnosed based upon the EM rash. The other thing that's interesting is that the bullseye rash that's become synonymous with the disease really only occurs in about 14% of patients. Again, this is a massive challenge in determining which patients are actually impacted by the disease. But emerging solutions are available, both commercially and in the research lab. And Jason Bobe worked with a series of Lyme experts and wrote a fantastic review on Lyme disease for the Frontiers of Medicine. And he categorized the emerging assays in 4 categories, which you'll see here, and we leverage in the paper. We couldn't unpack all 4 categories in the paper, but we were able to highlight a couple of promising techniques that could be transformative. The first is indirect detection. So antibodies are an indirect technique because they rely upon the host immune response. In direct detection, you're looking for evidence of current infection, either through DNA, RNA, or other molecules like proteins that are specific to the pathogen. Think of COVID. If you want to figure out if you have an active case of COVID, you don't get an antibody test, right? You get a PCR or an antigen test. The challenge is this has been difficult in Lyme because the bacteria are so elusive and they're in low quantities that are below the limits of detection of conventional techniques. But researchers at George Mason University have helped solve this problem by a couple of ways. One, leveraging the fact that Borrelia tends to colonize in the bladder, and then 2, using sample concentration techniques to increase the sensitivity. With these methods, they were able to show 100% sensitivity and 98% specificity for acute Lyme using the OspA antigen in urine. They were also able to confirm active infection in chronic post-treatment patients, demonstrating that infection persists after treatment. And I'll come back to this in the treatment section. So the team at George Mason continues their research and were recently awarded a grant through the Lyme X Challenge. This assay has also been made commercially available by Galaxy Diagnostics, and this is one of the many reasons I decided to join the company. Another novel approach being developed at Duke University, and it's actually in the lab that was started by Dr. Neil Spector, and his goal was to bring the tools and techniques of cancer research to the Lyme world. And this is actually based on an imaging modality. So here, researchers target a highly conserved and specific protein called HTPG, which is expressed on the surface of Borrelia cells. They then bind imaging agents to the HTPG protein and then can selectively label the bacteria for subsequent detection. So this approach has been demonstrated in a mouse model using fluorescence, but future work targets combining the technique with PET imaging so that they can pinpoint the location of infection. So it's easy to see how this could be transformative in complex cases to establish causality and the linkage of the bacteria to disease states and to understand the complex systems and what's caused behind them. So next is the link to chronic conditions. We've established that Borrelia is elusive. And another thing to understand about Borrelia is that it can travel throughout the body and cause conditions across multiple body systems. The link to dermatologic systems and rashes has been pretty well established, and there's also a lot of literature about Lyme arthritis and how the infection can lead to joint pain and swelling. But 2 key body systems where more research is needed and to understand how this bacteria links to disease are the brain and the heart. Now, we know we have neurological Lyme and that there are severe impacts. I've seen this firsthand. We also know that Lyme carditis is severe and leads to tragic outcomes, as it did for Dr. Spector. But with the massive misdiagnosis rates that I discussed in the last section, and with new research in this space, it's become clear that the story might be bigger than traditional medicine may imply. So first, I want to talk about the brain. The blood-brain barrier is a layer of specialized cells that are supposed to protect infection and other insults to the brain. But scientists have known for over 100 years that this barrier is not infallible. So Borrelia is in a class of bacteria known as spirochetes. And spirochetes were first discovered in the brain back in 1913, when a Japanese scientist identified the spirochete causing syphilis within brain tissue. Borrelia is also known to cross the blood-brain barrier, and this has been documented in the literature by multiple methods, including culture, PCR, and labeling in tissue sections. In fact, the figure on the right shows fluorescently labeled Borrelia in the brain of a rhesus monkey. And note that these bacteria were actually found after treatment with the standard course of oral deoxycycline, which is a thought that we'll return to later. There hasn't been a systematic study of what happens in the brain and how Borrelia leads to disease, but we do have smatterings of findings in the literature over the past 3 decades. Fortunately, I didn't have to wade through 30 years of medical research because the panel of experts in the Federal Tick-Borne Disease Working Group did that in 2022. They found a host of case reports and studies linking Lyme to psychiatric manifestations and cognitive issues, and recommended that Congress fund additional studies to understand these linkages and to determine the link between Borrelia and these costly conditions. Just to highlight a few key findings, in 2021, Dr. Brian Fallon from Columbia University used a nationwide Danish registry to determine if patients with a hospital-based diagnosis of Lyme disease had an increased risk of subsequent psychiatric disorders. He found that people diagnosed with Lyme had a 28% higher rate of any mental disorder, a 42% higher rate of affective disorder, 2 times the higher rate of suicide attempts, and 75% a higher rate of death by suicide. With the growing mental health crisis in America and worldwide, this is a critical area requiring further study. Another condition that's near and dear to my heart because of my husband's condition is dementia and Alzheimer's disease. Clinical case reports and autopsy studies have linked Borrelia to dementia for decades. In fact, one of the silver linings of COVID is that the scientific community has now embraced that chronic infection can lead to cognitive decline. And in fact, research at Harvard University demonstrated that the amyloid beta plaques that have become synonymous with the disease are actually a response of our innate immune system. So the question is, a response of the innate immune system to what, right? Usually that's an infection. So the question is so intriguing that a consortium of scientists called for an investigation of the link between key microbes, including Borrelia, and Alzheimer's disease. Next is the heart. So we talked about Dr. Neil Spector, and it's been well documented that Borrelia can infiltrate the connective tissue around the heart and cause Lyme carditis. This condition is severe, and atrioventricular or AV block is the most common presentation and leads to high-degree heart block in about 2/3 of cases. If untreated, the condition leads to pacemaker implantation and even death. Lyme carditis is generally thought to be rare, with an incidence of 1.5%, often cited in the literature, but it's important to note that this is a percentage based upon a specific subset of CDC-reported cases, and so the inputs into that percentage are pretty limited. On the other hand, high-degree heart block is one of the most common conditions leading to the implantation of nearly 250,000 pacemakers annually. And half of high-degree heart block is caused by what's known as idiopathic fibrosis. Idiopathic is one of my favorite words in medicine because, as far as I can tell, it's a fancy way of saying that we have absolutely no idea what's causing your condition. So the question is how many of these may be related to Lyme? And in a 2022 study of 130 patients with idiopathic AV block were tested for Lyme disease. 23% showed antibodies for Borrelia, and 12% of those showed antibodies consistent with recent infection. So 23% is a pretty big number, especially when we know that the tests have some issues and are not extremely accurate. So the question is how many cases are linked to infection? We know that we have 10 million Americans suffering from chronic mental illness. We have 6 million Americans suffering from Alzheimer's, over 3 million from heart disease. How many of those cases are linked to infection? And an infection that could be treated and cured and all of a sudden remove these people from the chronic disease registers. So even if it's a small percentage, the implications for the healthcare system could be huge. I know the implication for each individual family is life-changing. So finally, we have treatment. And I'd like to point out that I'm definitely not a doctor, so I will keep the discussion to literature and guidelines, and hopefully Jackie can help with any questions that we have in the Q&A. So One thing that's clear from multiple studies, only one of which I highlight here, is that it's easiest to treat and clear the infection when it's diagnosed early. The literature refers to Lyme disease in different stages: early localized disease, early disseminated Lyme disease, and then late disseminated disease. And the later it gets, the harder it is to treat. And patients with delayed time to treatment, and delay here is not long, it's only about over 30 days. Those patients are over 2 times more likely to develop persistent symptoms. So with the diagnosis statistics that I highlighted earlier, this is a tough finding to hear because there's so many factors working against an early and accurate diagnosis. So what are the recommended treatments? Well, that's not a topic that we can unpack in this webinar, but the IDSA treatment guidelines, by their own admission, leave huge holes. So this slide shows a summary of the recommended guidelines by clinical presentation and the grade approach that they use to rate those recommendations. Note that high quality evidence is lacking for all disease states, and several disease manifestations have weak recommendations with low or very low quality evidence. So this is precisely the reasons why organizations like ILADS exist: to share best practices among clinicians and to rapidly disseminate information to help those patients that are suffering. So what happens when patients are treated with the standard guidelines? Well, in 2022, a study out of Johns Hopkins University showed that even in ideal condition, treatment fails a significant percentage of patients. So what do I mean by ideal? Well, they looked at 234 patients who presented with EM rash. So they knew we have Lyme because they have the traditional EM rash, and then they received timely treatment with deoxycycline. Of those 234 patients, 43% experienced persistent symptoms, and 14% experienced persistent symptoms that impaired their quality of life. So these known treatment failures have led to the research case definition known as post-treatment Lyme disease, or PTLDS. And studies estimate that there are over 2 million Americans that are living with PTLDS. So PTLDS and persistent Lyme has been a subject of debate, and several potential causes have been suggested. I've included a list here with references, and they include many different mechanisms. They include infection-induced immune dysfunction, autoantigens and central nervous system sensitization, inflammation due to persistent Borrelia, inflammation due to Borrelia bacterial debris, and inflammation due to other infections. So it's important to note that that these mechanisms have been sufficiently supported in the research as a possible mechanism, and that the likely answer isn't just one of these causes. It's actually likely that different causes occur in different patient groups, and that multiple causes can actually occur within the same patient. But of that list, persistence of Borrelia after antibiotics has been hotly debated in the research literature. Through the work of talented researchers at Tulane and other top centers, I would say that there is now overwhelming evidence that Borrelia does persist after antibiotics. This has been demonstrated in cell cultures, animal models, and human studies, and I list many of the references here. One notable finding is shown here in the pie chart. So this study uses the nanotrap antigen assay that I referenced earlier in the diagnostic section. This study looked at 100 PTLDS patients who were all CDC criteria negative. And antigen testing revealed that 41% of these patients were antigen positive, implying that a significant percentage, but not all, of the patients with PTLDS were dealing with persistent infection. Others may be dealing with other causes listed on the previous slide, but further research is needed to truly figure that out. And several researchers have looked at alternative therapies. That imaging technology that I showed earlier out of Dr. Spector's lab at Duke, well, instead of binding HTPG with an imaging agent, they've demonstrated that binding that with a toxin will selectively bind with Borrelia and trigger cell death. So the small molecule that they call HS291 has been demonstrated in cell cultures and is now moving to animal studies. Other researchers have looked at antibiotics and combinations of antibiotics, which have shown effectiveness in eradicating Borrelia in cell culture and animal models. So the lower study on the graphic was actually not referenced in the paper since it was just released last month after we issued the paper. But it's a notable study out of Tulane University showing that 4 dual combinations and 3 triple combinations of antibiotics effectively eradicated persistent Borrelia infections. This study is also notable since the authors went out of their way to confirm clearance through multiple testing modalities, since, as we've discussed, testing is a challenge. So the moral of the story on the state of Lyme research is that we need more research. This area of science has been dramatically underfunded, and the implications are likely to expand across the healthcare system and many different disease states. So with the release of this paper, CLA challenged Congress to allocate $500 million to $1 billion per year for tick-borne disease research. Because we believe that there's many promising solutions out there, but more funding is needed to bring them from the lab into the bedside. The topics in the paper were broader than what we covered today, but the core recommendations are shown here. We need to increase research funding for diagnostics to get a sensitive and specific assay that can actually detect Borrelia and other tick-borne infections. We need to look at alternative therapeutics to treat acute, late-stage, and persistent Lyme disease. And then we need more clinical studies to better understand the mechanisms for Borrelia persistence and its tolerance to antibiotics. Finally, research needs tools. And so one key recommendation is to build a national biorepository of human samples for Lyme disease and other tick-borne illnesses. So with that, I'll pass it back off to Jackie and Bonnie, and we can talk about how you can help CLA to drive this change.
That was so great, Nicole. Thank you. And now we'd like to introduce Ms. Bonnie Crater, who will provide an update on the Center for Lyme Action's legislative activity. Bonnie is the co-founder and board director of the Center for Lyme Action. Bonnie's also co-founder and co-chair of the Bay Area Lyme Foundation and the former chair of the BAL Science Committee, which drives the research agenda, champions innovation, and seeks partners to chart new paths in Lyme research. She currently serves on the board of the CB Wellness Foundation and on the Town of Portola Valley's Nature and Science Committee. Please welcome Bonnie Crater. Thank you, Bonnie.
Thanks, Jackie. And I'm going to share my screen. Here we go. And get my slides going. That's our website. Okay. All right. Well, thanks everybody for joining. And I also want to thank Nicole for doing a terrific job on this very difficult paper to write called The State of Lyme Disease Research. If you'd like to read the paper, just go to our website, centerforlymeaction.org. And on the homepage, you'll see a button that you can click, and then you can go to the site where you can download the paper. It is a bit long, so apologies for killing a few trees if you decide to print. So my journey with Lyme disease started about a dozen years ago when my friend Lori Woods and I decided to start a foundation called the Bay Area Lyme Foundation. And that foundation is the #1 public charity funding Lyme disease research in the United States. But $45 million raised and 150 research projects funded later, we've realized that we needed to do more. We actually, we felt that we needed more money to come into this field in order to fund the research agenda that was really required. And so the idea for Center for Lyme Action was born. So I'm going to talk to you a little bit about the organization, kind of how we do what we do. And if this is your jam, I invite you to participate. So this is the world that we know that we live in. The Lyme cases are increasing dramatically. We all know this. We also know that the NIH funding, which is the primary funder of most clinical health related research in the world, actually, that funding for Lyme disease is actually quite low. You can see here in this chart that Lyme disease, the per patient funding for Lyme patients is $82 per patient. While the funding for malaria is $150 per patient, and West Nile is $6,870 per patient. So we have a long way to go to get proper funding for this area. And we desperately need it because we don't have really good diagnostics, and we struggle with therapies, particularly for patients with persistent Lyme disease. So Center for Lyme Action was born to address this issue, to really harness the power of the federal government, to harness all that money that's out there that we should be and can take advantage of. Center for Lyme Action is a new kind of nonprofit. It's a 501c4. And a 501c4, of course, is a special nonprofit where you're allowed to do unlimited lobbying of the federal government. Our mission is to grow federal funding for Lyme and tick-borne disease. So we're really focused on the money. That's where we're very heavily focused. Our strategy is to leverage professional lobbyists to advocate for Lyme and tick-borne disease research in both the legislative and executive branches. So we're trying to use, and we do use, tried and true tactics that do work for lobbying the federal government. Now, our organization shows up in a certain way, and I want to talk to you a little bit about that. These are our values, and values are very important because it is how you show up. We are bipartisan, and as our friend Kristen Honey says, the ticks don't care if you're a Democrat or Republican. This is a bipartisan biting situation. So bipartisan is the way we show up. We try to be as inclusive as possible because there's a lot of different opinions about Lyme disease, Lyme and tick-borne disease, and we want to try to include all those points of view as much as we possibly can and still move the ball forward. Now, there's a lot of sick people out there, so we need to operate in a very humane way and really make sure that we're being very conscious of how we operate so that patients can also participate in what we're doing. And we also believe in science. We believe that science will solve this problem. Now, our organization is member funded. I just want to highlight and give a shout out to a few people and individuals, as well as organizations that are our funders. Alex Cohen, the Bay Area Lyme Foundation, Project Lyme, the Fairbairn family, Lori Woods, and the Driscoll Family Foundation. Without these key funders, we couldn't operate and do what we do. So, okay, so what have we done? So the organization's been in place since 2019. And we actually have accomplished quite a bit since we started. So here's the chart, which shows the Lyme and tick-borne disease funding, federal funding. We started off at $59 million of federal funding. And now you can see there's been a nice progression over the last 3 years, almost tripling the amount of funding to 178.5. And where this money goes is it goes to three primary places right now. First is the very important research program agenda for the NIH around Lyme and tick-borne disease. And that funding, when we first started, was around $26 million a year. In fact, the funding was actually declining in real dollars. So we're excited to see, that funding going up, by 3- or 4-fold to $119 million. The second area is the CDC, which is currently funded at $52 million. And then there's a great program in DOD called the CDMRP program at $7 million. It's a congressionally directed mandated research program, which has a tick-borne disease research project inside of that program. So these are all the places that get funded. And as a result, some very interesting things have happened. When you increase the funding, it allows the government to do a lot more work and work on our behalf. So we're very excited to see, many of you may have seen this in July of that the NIH had awarded 5 projects. These are R01 grants, they're large grants, to 5 researchers that are super deserving, and many of you may know them. So these are the researchers and the institutions that were funded. And we're excited to see this kind of thing happen because we've never seen a series of R01 grants like this for post-treatment Lyme disease research. So this is really great. So R01 grants are a 5-year program. The first year, the awards were $3.2 million. So if you multiply that times 5, it's over $15 million over 5 years. So that's pretty exciting. Okay, a little want to share with you a little bit the method to the madness here, which is how do we do it? This is the regular appropriations schedule. So this is what happens in the federal government when the government is planning the budget. So the president puts together a budget that's usually released in February. Then there are budget hearings in March and April. Then there are these markups, which are to the bills, the appropriations bills. There's sometimes a vote on the floor in June and July. Then all this is happening, some on the Senate and some on the House side. The House usually goes first, the Senate goes second, usually, not always. And then we'll have a conference for their bills, and then the appropriations are signed into law. Now, this is a regular order, but obviously you've seen the Congress functioning in different ways over the last, well, when I've been really paying attention to the last 3 or 4 years. But in general, this is the order of operations. And what we do is we mirror and have activities for every step of the way. So in February, we have a fly-in. And at the fly-in, we set up meetings with people from all over the United States, Lyme advocates from this last year from 46 states across the United States to meet with their senators and members of Congress to talk to them about federal funding for Lyme and tick-borne disease. We then follow up with those meetings to fill out a lot of forms, so constituent appropriations requests. We then write a bunch of letters and have some face-to-face interactions. And then hopefully by the end of the year, we were able to celebrate, which we have been for the last 3 years. So just a little bit more about our fly-in. So we do it, the first fly-in that we did was in person in Washington, D.C., but then COVID hit and we went virtual. So we've been doing these fly-ins by Zoom, which has worked out really well because we have patients that can't travel to Washington, D.C. be able to participate, which is great. So the task is actually quite enormous. Last year, we had nearly 350 people sign up. We set up 244 meetings. We had 46 states represented. And then afterwards is the part that's really important after the meetings is actually submitting the appropriations forms to the different Senate and House offices. So last year, we submitted 1,458 appropriations forms to 235 Senate and House offices. So this is an enormous task and is critical as a critical piece of actually being successful. We're also really excited about the fact that we have that this activity can participate by all sorts of folks in all sorts of Lyme organizations all over the country. And we have more than 40 independent Lyme organizations that are active in Center for Lyme Action, and over 2,000 folks on our mailing list that are actively doing things, signing letters and meeting with people. So just to wrap it up, if this is something that you think would be fun, and it is actually pretty fun, talking to the Senate and House staff and sometimes members of Congress, this is how to get involved. So what you can do is just go to our website at centerforlymeaction.org and scroll down to the bottom of that homepage, and you'll see a pop-up, and you can enter your name and your email address, and you'll get on our mailing list, and you'll be notified of different things that you can do. You can become a member. So again, going to our website, we are a member-funded organization, and so becoming a member is super helpful to us. There's individual $30 memberships, and then there's memberships for organizations. So you can click on our membership tab. And then lastly is we've just opened registration for our 2024 fly-in. So our virtual fly-in, which will be held on February 20th and 21st. And you can go to our centerforlymeaction.org events page and sign up there. So thank you very much, Jackie, for hosting us. And we're thrilled to be part of this webinar.
Yes. Thank you, Bonnie. I really enjoyed your talk and learning more about legislative process, and you as well, Nicole. This is just such a great opportunity. Thank you both. We do have several questions from our audience in the Q&A section. If you have any questions, please put your questions there. Let's go ahead and open those up. A lot of thank yous. Yes, thank you very much. Can anyone present or give any insight or updates related to venom therapy? Recently, extensive research at Johns Hopkins and Tufts, bee venom component. Is it effective at killing various types of cells? I know not necessarily related to Lyme, but I know of any research related to that? I know a little bit about it, but not enough to answer that
Yeah,
question.
unfortunately, I don't know enough of that research either to talk about it in any detailed manner. So I think that what we focused on was some of the more traditional things that are out there, but there are definitely lots of alternative therapies where, the research determines what is effective in which patient groups.
And then hygromycin A. Do you know any? Let me answer the question about the bee venom. It's always about the dose, right? The question is, we know that it certainly works with some patients in some types of treatments. It's always about what dose is the most effective for whatever treatment it is that you're looking for, whatever disease state. So it always comes down to the dose. And I think that's what they're looking for right now to answer the bee venom question. The hygromycin A, am I saying that right?
Yeah, so we did, hygromycin A. So we did reference a study that was shown in mice that actually showed clearance of Borrelia. And I think that, again, with all of the studies that we have on antibiotics and combination antibiotics, right now they're limited to either cell cultures or mouse models. And then and how you do and what you do in mouse models can vary pretty significantly based upon the research lab. And so, but the results show some promising results. And I think it warrants further study and investigation.
Another question about Galaxy test. Is it covered by insurance?
Some insurances do cover, yes, partial. Absolutely.
Got it. And you can get that from your medical provider, or can anyone order that test?
Yep, from your medical provider. They can go online to the Galaxy website. There's all sorts of information about how you can get information and run testing.
Got it. Perfect. Okay, question for you, Bonnie. What exactly happens at a fly-in? I was wondering that too.
Yeah. So what we do is the main event is having a meeting with your member of Congress and your 2 senators. That's the main event. And what the object of the game is to pitch to these folks the problem of Lyme disease, to educate them further, as well as areas where they can make investments to help us solve the problem. And so what we do at Center for Lyme Action is we match up all the folks that sign up for the fly-in, we'll put folks into groups, and the groups chat with the member of Congress or their staff. It's usually the staff. The staff is most important because they actually are the ones that are really writing the legislation and have a key influence on what actually happens. So to prepare for this, we prepare materials and we do a training session the day before. We also do an optional training session the week before. And we're adding a new program this year to have team leads for various regions to help folks with preparing for the fly-in. But it's a lot of fun. You get to meet new people that you haven't met before and talk to your members of Congress or their staff.
I have another question for you, Bonnie. This is a great one. How do we advocate? Because we're all constituents of somebody, right? How do we advocate health insurance to pay for Lyme treatment? How do we go about that from a health policy standpoint?
Yeah, it's very tricky because there is disagreement about about diagnosis, right? And so that's why we've been focused on research to create a good diagnostic and therapies that are proven as well, and proven in the standard medical community as well. So we're kind of at step 1, which is we need a good diagnostic, basically, so we know who's sick and who's well, and know that reliably in order for us to turn on the healthcare system.
If I could point a point there as well, is if patients communicate to their providers, their medical providers, about ILADs, that we exist, being able, there was a question here about, how do we teach doctors about Lyme disease, right? It's really about grassroots, getting the medical providers to understand it, invite them to labs. Hey, I want you to go to iLads, learn more about Lyme. There should be some sort of an outreach there as well. I know about clinicaltrials.gov, but we have a question here about how to be a part of your research trials. Miss Bell, how do they get a part of any of the research trials? I know I used to do the one with NanoTrap. We did that at my previous office. But how do patients get into these research trials?
So we don't have at Galaxy, we don't have any research trials that are currently open. We do support testing for researchers who are, in universities. We'll support testing. And if there's anyone that wants to use our services as part of their test study, we're happy to engage on that. I think as we advance, we will be opening up some trials, and generally we would announce that on social media or other places as ways to get involved.
Okay, perfect. There is a question about research on the use of methylene blue and tick-borne diseases. There is information. I know at iLads, there was a great presentation at the last conference about methylene blue, where we talk about it quite a bit as medical providers. Do either of you have anything to mention about the research and about the treatment with methylene blue and Lyme disease?
I do not. I mean, I really focus the paper on things that are in the scientific literature. And so there's always a little bit of a lag time, based upon what people are seeing in clinical practice. And so that's why I think, forums like iLads and the meeting are really great, because then you get people that are boots on the ground. And some of, a lot of people are busy doctors. They don't have the ability to do published research. But so this paper was really more about what's been published, and then trying to get more funding for running more clinical studies and engaging more doctors to get that information. Because at the end of the day, that's what it's going to take to drive the system, to drive NIH, to drive the healthcare system. You need that scientific data collected in a systematic way.
And just one quick comment on that as well is any of the physicians on the phone who have time and can write a case study on some of these topics, it would be terrific to get that publication. Those papers are usually a little easier to put together than obviously a clinical study or something like that.
Yeah, I mean, that's a great point. So a lot of the papers that I reference in the section on Alzheimer's and psychiatric symptoms, those are case studies, right? Those are things where people found an interesting case. They published what they found. In some cases, they included autopsy results, really showing, some of the indications. That's where things start. I'm an engineer, as you mentioned at the beginning, and it always starts with an N of 1, where the number of samples equals 1. And that's what that case study is. So it's very important to get those stories out there.
Question regarding Galaxy, is the urine antigen test, is that available in New York State?
It is not at this point, but that is definitely on our radar screen.
Got it. Another question about the fly-in, Miss Bonnie. Will there be one that's virtual in DC, or virtual that other people can attend?
Yeah. So
if they can't be here.
Yeah. So our fly-ins are all now virtual. We decided to go virtual in the pandemic, and we're staying virtual. And the reason is that it allows a bigger group of Lyme advocates to participate.
Yeah. Good. Just to let everybody know, we cannot answer personal questions about your treatment or testing or diagnoses. Just, I want everyone to know that, why I'm not bringing that up. But there are multiple comments about requesting the video, which I know will be posted online, and a copy of the slides. Not sure if that will be available as well. How long does a tick typically have to be attached to a person before transmitting Lyme? It can be, my answer is it can be pretty quick, right? It doesn't have to be minutes, hours, days. It can be pretty quickly for not just Lyme, but all co-infections. Today, we're just talking about Borrelia burgdorferi. But for all bugs, it can happen within moments. And you may never know the tick is attached. Do you have any other comments, Ms. Nicole or Ms. Bonnie?
No, that wasn't, As I said at the beginning of the presentation, this was a daunting project to summarize all Lyme research. And so, we weren't able to cover every topic. We really kind of focused on things that were, the top areas where we knew we needed a little bit more funding, so I did not personally dive into that part of research, but I know that there's been a lot of studies that have been done on that.
Got it. Another question, legislative question, Ms. Bonnie. What is being done on the legislative level to get more labs that offer medical testing with more bans like the Vibrant America Tick-Borne test recognized by the FDA? So
Okay.
how can we get more testing recognized
Yeah.
by the FDA and covered by insurance? I guess it's the definition and the discovery of Lyme.
Okay, so this is a complex topic of diagnostic companies applying for certification, and there's kind of two paths. One is to do a laboratory developed test, which is what's called a CLIA certified lab, and those laboratory tests are tests that you can order and have your doctor can order them. Sometimes patients can order them directly, but it's done in a lab, and those labs are certified by actually by a division of Medicare. Then and Nicole should be answering this question, but anyway,
You're doing a great job.
Okay, the second path, right, is FDA certification, and diagnostic tests are considered a medical device, and so you get a certification called a 510K certification. Now, this is a very complex certification that requires a lot of expense, work, and so on, and it's very difficult to get. And so today we don't have very many Lyme tests that are FDA approved. We have very, very few. That the majority of the action that is happening in laboratory tests. One of the advantages of laboratory tests is that it allows you to evolve the test over time. If you get 510K certification, then it's very difficult to log. Every time you change the test, you have to go back and get recertified, and that's very expensive. So in these early days, right now, there are most of the work that's being done right now is in laboratory developed tests. So there's nothing that I can do to get companies to apply for FDA approval. But if you run a company and you have if you have a diagnostic test and you want to get further certification because you've got a test that is fairly stable, you can apply to the FDA and get 510K approval.
Got it. How much funding, another legislative question, how much funding actually goes into prevention vaccines? Do you know?
It's a small amount of money. If you go onto you can do this yourself. You can go onto the NIH spending website, and you can search for Lyme disease or search for tick-borne disease, and there's a listing of all the projects that are funded. And if you download that spreadsheet, you can see that there's projects of vaccine that are dedicated to vaccines. And in fact, Nicole did this as part of her research paper. And I don't know if she has that page open.
I don't. I was trying. Like I can go on my computer and get it.
Yeah, but it's a very small amount. But as everyone knows, there is a vaccine that's in phase 3 trials. That's Pfizer and Valneva's vaccine test, and that's an ongoing project. And you can follow the gory details on clinicaltrials.gov and in the news.
Yeah. And as Bonnie mentioned, in the paper, we looked at the 2022 numbers in Lyme disease, and we kind of categorized it by the different areas: prevention, diagnosis, treatment. And so you can see the breakdown of the funding in NIH in each of those sectors. I don't remember the numbers offhand, so I don't want to misquote them, but they're in the paper.
Another great question, kind of combining legislative and treatment, is, I find physicians relying on CDC guidelines. Should patients be referred to Lyme specialists for treatment? Because, and there was a question earlier about the CDC guidelines. Why do we keep giving money to the CDC or keep trying to do this? But it's all connected, right? It's all connected. How do you, many providers look to the CDC for guidance for many different health issues. How do we change that paradigm?
So one of the things that's very true is that the government can't do anything new unless we get them more money. Like nothing new will happen. And that's why we're focused on growing federal funding for Lyme and tick-borne disease, is because that will allow the government officials and people that run these departments and do new things and fund new projects like those persistent Lyme disease projects that were funded by the NIH. So this is by increasing federal funding, we're actually creating change.
Got it. Fantastic. I can answer this question. Can you speak to the connection between chronic inflammatory response and chronic Lyme? Chronic Lyme is a segment of chronic inflammatory response, also known as CIRS, typically related to the interior of a water-damaged building. Think mold, actinomycetes, Corynebacter. We think of Lyme. We think of, different types of psychiatric PTSD-related illnesses, all related to CIRS. It is a segment. And there is a genetic test, an HLA, human leukocyte antigen test, to be done to see if you are in that roughly 25% of the population that does have that genetic sequence. That is covered by lab. It's done at LabCorp, covered by insurance. Your medical provider, any medical provider, can order that test. Again, it's available for everyone. Again, everyone, we cannot answer questions based on your personal questions about treatment, what medications to take, what have you. Miss Nicole, you briefly mentioned azlacillin in a flyer. Any more information that you could provide?
No, not that I would be comfortable sharing on the call. I have the reference is in the paper, to the paper, to the paper that I referenced in our paper. And so I would encourage people to take a look at that to see what the published state of knowledge is, and then we'll continue to monitor it as we go forward. Bonnie's already planted the seed that she wants me to do some sort of update to this as more research emerges. So we'll hopefully be able to do that as things go forward.
Yeah, and if you have questions about azlacillin, and if I would encourage you to read the recent publication from Monica Ember's lab from Tulane, because it does refer to azlacillin, and azlacillin as a single antibiotic was not proven to eradicate Borrelia in the long-term chronic mouse model.
How do we approach our medical providers who are dismissive with information to get them on board to believe all that suffer from this illness? I would say give them Nicole's paper.
Yeah, I mean, that's one of the things that I'm hoping people will be able to do. Is it's not just for Congress, but it's also for the general public. And so if you have, first of all, if you have a medical provider who's just completely not open to it, then find another medical provider. But if you have a medical provider that wants to, learn and grow, I hope that the tool is the paper is a good tool for people to get a good summary, because it is a complex landscape. There's literally, hundreds of papers that are relevant that we reviewed. And so I think that, hopefully this provides a little bit of insight without having to go and read all those. Something that I was able to do on my own, right? This was on the back of the Federal Tick-Borne Disease Working Group. It's on the back of the NIH strategic plan. We also had some experts that helped us review this paper and make sure that we were using the latest research. And so I want to thank them for all their work to help make this a reality.
And there was actually 3 more questions about the fly-ins, Ms. Bonnie. So that's a hot topic in this.
Good. Yeah, please come. Go to our website, sign up today.
They want to do someone had said, or have you considered conducting fly-ins with associations, boards, medical providers, just versus everybody, just focusing it on medical providers and.
Yeah. Well, I think medical providers is the purview of ILADS. It's a fantastic organization where any medical provider can learn a lot about Lyme and tick-borne disease. So I would refer any medical provider to ILADS and go to the conference and participate in events to learn more.
Someone had mentioned that about different testing. I know in a question, someone mentioned a company that I said out loud. We do not endorse any companies, right? Any ILADS does not recommend one company or another over testing, right? We do not talk about which one is better or potentially not as good. So I apologize if that was misconstrued. We do not encourage any particular test. But you can talk about that with your own medical provider. Please ask all those questions to your specific medical provider. Is it possible that some people can fight off these infections without needing treatment?
I mean, I think that for certain tick-borne infections, there's evidence that they can, and I think that there's also evidence of asymptomatic infection. When you look at some of the literature of antibody, prevalence in people that are, non-symptomatic, how that occurs and whether they develop into symptomatic cases later on is something that requires further study. I mean, I know, for example, in my own personal case, I know my husband was infected likely for an extremely long time before his condition actually evolved. But that's a personal experience.
Yep. Let's see. I think we are out of time. So, everyone, thank you for joining us tonight. We have several virtual learning opportunities coming up that you may be interested in. Our next webinar will be on December 20th with Dr. Tom Moorcroft talking about tenefriquin. And in January, we'll be hosting a Saturday morning virtual seminar on environmentally induced illnesses. I will be speaking at that one. Registration for all these events can be found on the ILADS website, www.ilads.org. ILADS is a professional medical society dedicated to the diagnosis and treatment of Lyme and other complex inflammatory diseases. Membership is open to medical clinicians and scientific researchers. You can learn more about membership opportunities on our website or email us at membership@ilads.org. Thank you again for joining us, and please take a moment at the end of our broadcast to complete a brief survey of tonight's presentation. Thank you.